All right. So this is the version that I'm doing for those of you who couldn't make it to one of the live sessions. I'm just going to work through osmosis, pretty much not going beyond anything that was not in the textbook. So reading the textbook is pretty much giving you the same information, but you're getting my emphasis and explanation through this video. So tomorrow we're going to be doing an osmosis lab, as long as I can get into school and make the solutions. And just want to review again what concentration gradient means, because you need to understand that to understand osmosis. So you should have out your active reading guide for 5.3 and the section on osmosis, diffusion of water through a selectively permeable membrane. So just reviewing again this idea of concentration gradient. In this picture, there's a higher concentration of these orange solute molecules than on the other side. This is also a concentration gradient, not as steep as this one. But what will happen is molecules tend to move down their concentration gradient. They tend to spread out to get to that dynamic equilibrium that we've talked about. Now, this molecule can actually pass through the membrane. When we talk about osmosis, we're going to have solutes, ions, or polar molecules, things that can dissolve in water that cannot pass through the membrane. And then that's going to affect the direction that water travels. So again, have your 5.3 active reading guide out as we work through this information. So this slide is pretty much showing you the phenomenon, and you need to understand why water is moving more to one side of the membrane than to the other side of the membrane. So first thing is, osmosis is the diffusion of free water molecules across a selectively permeable membrane. I'll explain more what I mean by free in a moment. But let's look at the setup. So we've got this U tube. It's a tube in the shape of a U. And on one side, we have placed a solution with a lower solute concentration of sugar, let's say sucrose. We have a membrane that is impermeable to the sucrose. The sucrose cannot diffuse down its concentration gradient. If it could, the sugar molecules would tend to move to this side more than the other side, so high concentration to low concentration of sucrose. But there's a membrane here that prevents the sucrose from passing, just like our dialysis tubing tomorrow. And then on this side, we've added the higher concentration solution. And it's always comparison to the solution on the other side of the membrane. So what will happen as time passes is that water will move to this side faster than it moves to this side, and this would be the end result. So the water levels are now no longer even, but the concentration of solute on both sides of the membrane is even. So most basic thing to remember is that water will always move from a lower concentration of solute to an area of higher concentration of solute. All the rest of this is explaining why that takes place, why that phenomenon occurs. So take a look at, I'm saying so a lot. Anyway, this diagram helps to explain why osmosis is occurring. Why, against the force of gravity, there is now more liquid on this side of the tube than on this side of the tube. And this would be actually a low energy state. So when you look at it a little bit closer, so we zoom in to that spot on the membrane, we see that the lower concentration of solute means that there's more free water molecules. So these water molecules are considered free because they can move on their own. They're not attached to anything else. Whereas these water molecules are actually not free. They are bound to this solute, what we're going to call a non-penetrating solute. So these holes in the membrane would not let that sucrose through. And any water molecules that are hydrogen bonding, forming that hydration shell around the sucrose, would be stuck on this side of the membrane with that solute. And then over here, we have a higher concentration of the solute. That means that there's more water molecules making hydration shells around those solutes, and there's less free water molecules. Water molecules free to move from one side to the other. So this whole osmosis thing is really an example of how memorization is not going to get you there. Understanding a few basic concepts and really understanding them and knowing them, and then being able to think through a thought process is how you're going to be able to answer questions and solve problems using osmosis. So we've talked about already that water will always move from an area of lower concentration to higher concentration. We've now talked about what that really means is lower concentration of solutes means there's less water molecules bound to the solutes, more free water molecules able to move from one side to another. So this is actually a really good mental model diagram right here. And higher concentration of solutes, ions, other polar molecules, will have more water molecules bound to those solutes, less free water molecules, less likely, just lower probability that a water molecule would randomly move out of this solution into this solution. With so many free water molecules over here with the lower solute, it's just more probable that a water molecule will move to the other side. And once it gets here, it'll form those hydration shells around the solutes. All right, so what does this mean in terms of cells? We're going to focus first on cells without a cell wall, and then talk about what happens with that cell wall surrounding the cell. So tonicity is the ability of a surrounding solution, so a solution outside of a cell, to cause a cell to gain or lose water. So gaining water, water would go in. Losing water, water would go out. And this tonicity, which can actually be quantified in terms of water potential, but this water potential due to solute, this tonicity depends in part on the solution's concentration of solutes that cannot pass through the membrane, these non-penetrable solutes. So if we go back to this diagram, these green things would be what we call non-penetrable solutes. So we'll start with isotonic. You should know the definitions of these words and then be able to walk through a thought process of what would happen to a cell if you placed it in these different solutions. So don't just memorize what would happen to the cell. Know what the solution is and then go through the thought process, the reasoning of why the cell, what the outcome would be if you put a cell in that particular solution. So our first one is called isotonic. Isotonic, iso as an isosceles triangle, iso means equal. The tonic is the concentration of solutes. So when we talk about tonicity, we're using words like these. So isotonic means that the solute concentration outside the cell is equal to, is the same as, the solute concentration inside the cell. So if you go back to this diagram, we would imagine that there'd be equal concentration, moles per liter, of these green particles on both sides, which is not the case here. Here it is. So these would be two isotonic solutions, equal concentration. And in this case, water molecules are moving. There's just as many free water molecules here as there are here. So water molecules are moving in and out at the same rate. So what did that look like in the cell? In the cell, water is moving in. Water molecules are just as likely to move out because the solute concentrations are equal, the free water molecule concentrations are equal, and the cell is going to stay the same. And they use red blood cells to illustrate this because red blood cells have this unique biconcave disc. So this would be normal. So let's go over here. That is definitely not normal. So a hypotonic solution, it means that the lower concentration of there's a lower concentration of non-penetrating solutes in the solution outside the cell compared to inside the cell. So these words are always a comparison. What's the solute concentration outside the cell, and is that higher or lower than the solute concentration inside the cell? Hypo means low, lower. Hypothermia, lower body temperature. Hypodermic needle, it's a needle that goes under the dermis of your skin. So hypotonic, lower concentration of non-penetrable solutes outside the cell than inside the cell. Going back to this diagram here, this would be the hypotonic solution. Less solute, more free water molecules to randomly move around. So you might guess that because there's more free water molecules here, they're more likely to move in than out. And so water would be constantly coming in. The cell would swell and it would burst. And this diffusion of water across a selectively permeable membrane is called osmosis. So osmosis is that water coming in. It's coming in because there's a lower solute concentration outside, more free water molecules outside than inside. And then over here, this is the hypertonic solution. So we've put a cell in a solution that has a higher concentration of non-penetrating solutes, ions, polar molecules. And it's higher outside the cell than inside the cell. So now this is the hypertonic solution. So we're saying that this is the solution outside the cell. This is the solution inside the cell. There's more free water molecules here, so they will move out of the cell versus into the cell. Right? So water is moving out faster than in. Water molecules out here are all around those solutes, making those hydration shells. They're just less likely to be moving on their own and go in. When the water molecules are in that hydration shell, they stick with that solute that can't pass into the cell. So the cell would shrivel, and that would be a bad thing to have happen. All right, so when we think of osmoregulation is the control of solute concentrations and water balance. You need to osmoregulate. Your kidneys are responsible for that. Also, on top of that, all of our cells are bathed in an isotonic solution. So in you, this is a solution outside of all your cells, which is isotonic. Same solute concentration as inside your cells, so water's going in and out at the same rate. That's one way we maintain osmoregulation. And then we also have our kidneys that help with that as well. Remember, our paramecium lives in fresh water, low solute concentration, so it's hypotonic out here. Water is constantly coming in, coming in, coming in, so they need that contractile vacuole to pump it out, pump it out, pump it out. And we saw the video of what happened when the contractile vacuole wasn't functioning. The paramecium swelled and burst. All right, almost done here. So this is now showing what happens when the cell has a cell wall. If you notice, information at the top is not changed from the previous slide. So hypo, iso, and hypertonic have definitions. These are the definitions. So when a plant cell is put into an isotonic solution, which would be normal for a cell without a cell wall, water will go in and out at the same rate because it's an equal solute concentration, equal number of free water molecules here versus inside the cell. But the cell will be what we call flaccid. The plant would look wilted. Not dead, not shriveled, but just kind of wilted, like you haven't watered it in a while. If we put it in a hypertonic solution, so a high salt solution like seawater or something like that, water will leave the cell faster than it's going in. The cell will actually, it's called plasmolysis. And this, you see the plasma membrane actually pulling away from the cell wall. This would eventually kill the plant pretty quickly. Early form of biological warfare back in the medieval times and before was dumping seawater or allowing seawater to flood your enemy's fields. The idea of salting the earth. It would make all the water outside the plant hypertonic, and water would not go into the plant. It would actually come out of the plant, and the plant would die. So isotonic and hypertonic, not so good for plant cells. Instead, hypotonic is great for plant cells. Hypotonic, lower solute concentration outside the cell than inside the cell. That means there's more free water molecules floating around here. So water will be coming in. The cell will swell, but it has the cell wall there to prevent it from bursting. So this is a wilted plant. This is a plant that's standing up nice and straight and healthy. And we call that, as the water comes in and pushes against that cell wall that prevents it from expanding, the cell expanding anymore, we call that turgor pressure, the pressure that the cell is pushing up against the cell wall, and the cells are turgid, and that allows the plants to stand upright and be a healthy plant. So isotonic would be homeostasis, proper solution for a cell without a cell wall, whereas plant cells need a hypotonic solution for homeostasis. So the rest of the assignment is looking through some videos and some tutorials on water potential. And water potential is really just a way that we quantify um, hypotonic, isotonic, hypertonic. Mr. Anderson will go through the formula for how to calculate it. We'll talk more about it in class. But again, the basic rule to begin with for all of this is water will always move from an area of lower solute, so here's the solutes, lower solute, to an area of higher solute. But it's comparative. Now this area has the higher solute, or lower solute. This side has the higher solute. So the water always goes from lower solute to higher solute. And we can actually give numbers to it. So this pure water has no solute in it, so the water potential is considered to be 0 based on the formula that you'll look at with Mr. Anderson's video. 0 is actually a very, very high water potential. All the rest of the numbers will be negative. This is a very low water potential, negative 50. And you're going to find out from the formula that this number is partially because of the number of ions or polar solutes in the solution. So water always goes, so this would be the most hypotonic. This would be the most hypertonic. But then if you compared this to this, this would be a hypotonic. If you compared this to this would be a hypertonic. It's a comparative term. Solute concentration on this compared to this. And you can see water always diffuses from the higher, 0 is higher than negative 200. Negative 200 is higher than negative 500. So it's all talking about why does this occur? So the way the lab is going to work is I'm going to make up a bunch of solutions of sucrose, ranging from distilled water, so 0 sucrose, to 1 molar sucrose. I'm not going to label them with their concentration. I'm going to make them different colors using food coloring. And your task will be to assign 0, 0.2, 0.4, 0.6, 0.8, and 1 molar sucrose to each of the colors. And we will hopefully do that in lab tomorrow, as long as I can get in and make the solutions. All right. So that is it. That was actually a little bit shorter than the half hour it was taking, because I had to make sure everybody was functioning. And that's it. Hopefully see you tomorrow. Bye.